Welcome back to this next video, and this is the part 3 on the drug designing. In the first part of the video, I've told you about the basics of the drug designing, and in the part 2 of the video, I've told you about the first step of the drug designing, that how you are going to find the target for an unknown human disease. And I've told you that when you are going to find a target, in most of the cases that targets they are proteins, and currently only 10% of the human proteins, they are known as druggable, or they are known to be druggable. Then I've told you about the properties of promising a drug target, and then I've told you about the how you are going to validate the a drug target. Now in this particular video, I want to focus on how to find a drug. Once you have identified a good target, a promising target, the next step in the drug designing is to find a suitable drug. Now let's say scientists have found the genes that code for a protein. For example, we'll call that protein as protein A, and that this particular protein that is involved in the unknown human disease. And the most important thing is that it can be targeted with a drug. That means that the protein A is responsible for that unknown human disease, and we can actually target this protein A. So this protein A that will be druggable. So how does one find a drug that will interact with this protein A? Now, if there isn't already a chemical known to interact with this particular protein, you have to go to test hundreds of thousands of chemicals quickly using robots in a process known as the high throughput screening. So when you don't know that there is a chemical which can interact with this particular protein, you have to go through the process of the high throughput screening. And in the process of the high throughput screening, you will be screening a large library of chemicals for their ability to modify the target, which in this particular case is the protein A. Now, to decide what chemicals to test, the medicinal chemists, they often use a computational technique called virtual screening that uses data from past screen to predict what molecules they are going to interact. So this is one of the important thing about the modern drug designing, that in the classical drug designing, you have to test them in the petri plates, you have to test them in the animals to know that which kind of the ligands or which kind of the drugs that can actually interact with your target protein. But in the modern drug designing technique, you do not go directly to the wet lab. You actually start from computers by utilizing this process, which is known as the virtual screening. And on the computer, you can actually check the interaction of the different kind of the targets with different kind of the drugs with this protein A. Now, once you have screened large library of the chemicals by the process of the high throughput screening, so the library of the chemical compound is then screened for interaction with protein A. And any compound which can interact with the target or the protein A in this particular case, that is known as hits. So from thousands of the compounds, the virtual screening may be telling you like 100 compounds, they are only interacting with the protein A. So only these 100 compounds that will be further screened for their different properties. Now, one of the other important function of this high throughput screening is that it is going to show you how selective the compounds are for the chosen target. And the ideal is to find a molecule which will interfere with only the chosen target, or that will only interact with the chosen target, and it is not going to interact with other related targets. Now, to this end, you have to run other screening tests to make it sure that whether the hit against the chosen target that will interfere with other related targets, and this particular process is known as the cross screening. When you talk about the living organisms, the targets, they may be related to each other, they may be related compounds or related proteins. In this particular case, there may be more related proteins to the protein A, so you have to choose that particular hit which only interacts with the protein A and not with the related proteins which share similarities with the protein A. And in this particular process is known as the cross screening, where you are actually checking the interaction of the hit with the chosen target and with the related targets. Now this cross screening is very important because the more unrelated target a compound hit, the more likely that off-target toxicity will occur with that particular compound when it reaches the clinical trials, and that will not be approved by the FDA. So the high throughput screening is actually going to screen thousands of the chemical compounds, and then that is going to give you a limited number of the compounds which actually interact with the target protein. Now, it is very unlikely that perfect drug candidate that will emerge from these early screening runs, from these early virtual screening runs. Now, it is more often observed that several compounds are found to have some degree of activity, and if these compounds share common chemical features, one or more pharmacophore that can be then developed. Now, what does pharmacophore mean? That pharmacophore are usually those particular compounds who have got steric and electronic features that is necessary to ensure the optimal interaction with a specific biological target or trigger or block its biological response. So once you have identified a hit, you can actually develop one or more pharmacophore. Now at this point, when you have selected a particular hit or you have selected a particular pharmacophore, at this point the medicinal chemist, they will attempt to use structure activity relationship, which is for short is known as the SAR, to improve certain features of the lead compound. Now I'll come to this, the difference between the hit and the lead compound in a while, but the medicinal chemist, they will attempt to use structure activity relationship to improve certain features of the lead compound. Now the lead compound is actually a compound that has pharmacological or biological activity likely to be therapeutically useful, but may nevertheless have suboptimal structure that require modification to fit better to the target. So these modification, they are actually determined by the structure activity relationships, and the medicinal chemist, they will actually improve the properties of the lead compound utilizing different features. Now, how they can actually improve the interaction of the lead compound with the target is that they can actually increase the activity against the chosen target by modifying the drug or by modifying the lead compound. They can actually reduce the activity against the unrelated targets. That means that they will modify the ligand or the lead compound in a way to increase its activity against the chosen target and reduce its activity against the unrelated targets. Now, one of the important things that the medicinal chemists do when they are focusing on the structure activity relationship, that they want to improve the ADME properties of the drug. Now, what this ADME means is, when you talk about the drug journey through your body, a discipline which is known within pharmacology, which is known as the pharmacokinetics. The pharmacokinetics actually tell you about the ADME properties of the drug. What this ADME mean is, it is anything but simple, that this A is short for the absorption, this D is for the distribution, this M is for the metabolism, and this E is for the excretion. That how a drug, when you take that particular drug, how that is absorbed in the body, how that is distributed to reach to its targets, how is then that metabolized in your body, and how finally that is excreted from your body. So what the structure activity relationship that is done by the medicinal chemist, what they do with the lead compounds is that they are going to modify them to increase their activity against the chosen target, to reduce their activity against the unrelated targets, and improve the ADME properties of the lead compounds. Now this process that will require several iterative screening runs, during which it is hoped that the properties of the new molecular entities that has been modified by the medicinal chemist, that is going to improve and will allow the favored compounds to go forward for in vitro and in vivo testing in the diseased model of choice. Now, the lead drugs that actually offer the prospect of being followed by the backup compounds called is the analogs. So the when you have selected the lead drugs, you can actually modify them in a way in which the target protein or the target epitope can modify itself, and these backup compounds, they are actually known as the analogs. Now, just like the validation of the target, you have to validate the hits as well. Now, validation of the hits that can be carried out in a variety of the ways, but at its core, hit confirmation depend on using a variety of biochemical assays to understand the interaction between the hit and the protein A. Now, most of the hits, they will unfortunately interact in a way that are not relevant to the unknown human disease, such as they will be interacting with a wrong part of the protein, and hence they will not be able to modify the function or the structure of that particular target. Now, any hit which survive this validation process, they are known as the lead compound. So you have got thousands or million of the compounds. From those, you actually choose only those particular drugs on only those particular chemicals which interact with your target. So those particular collection is known as the hits. When you validate those particular hits, and after validation, the compound which show better interaction with the target protein, then they are known as the lead compound. And the medicinal chemists, they actually they are going to modify the properties of this lead compound to give them a better chance to be approved by the FDA. Now, up to this point, one has spent millions of dollars and years of research, yet most of the hits, they will fail to turn into a drug because they will not be interacting with the target in a way so you can treat that particular unknown human disease. So when you talk about the difference between the hit and the lead compound, so a hit compound is a molecule that shows the desired type of activity in a screening assay. Now, lead compounds are selected from the collection of the hits by refining the screening criteria to enable the selection of the most promising molecules for further drug development. Now, how you are going to turn the hits into the drug? So one of the property was that the medicinal chemists, they will be going to improve the SAR and the ADME properties of the particular drug. Now, these lead compounds from the high throughput screening, they are like unseasoned, unprepared interacting molecule with your target. They may work somewhat well, but are often more useful as the foundation. Now, chemists have developed many tricks to create drugs that are more effective. Now, oftentimes, these tricks involve finding the right combination of modification to allow the drug to be more specific for its target. So, if you are making that more specific for your target, you are actually decreasing its interaction with the related or the unrelated targets, thereby minimizing their side effects. Nearly 100% of the time, the drug needs to be optimized to overcome many logistical challenges, such as surviving the digestive tract and being soluble in the bloodstream. Because if the drug you have selected, if they cannot survive the digestive tract, and if that is not soluble in the bloodstream, they will be having a problem in the distribution and hence reaching to its target. Now, furthermore, one must prove that the compound that will cure the disease or alleviate symptom in human cells and in the animal molecules. Now, this is the third step of the drug designing, and the most dangerous one, that is known as the valley of death. Now, what this valley of death refers to is that the when you talk about this term valley, that valley is actually the gap between the drug that work in the petri dishes and mice, where you have got everything in control, and the drugs that actually work in the human body, where you cannot control anything. So the valley of death actually is the gap between the experiments that you perform in the petri dishes and in the mice. And when you perform the experiments in the petri dishes and the mice, that is collectively known as the preclinical stage. And the drugs that actually work in the human body, you actually check this in the clinical trials. We will talk about that in a while. Now, the main obstacle for candidate drugs is toxicity in clinical trials, because when you introduce that particular drug into a whole organism or living organism, then you are going to have these toxicity problems. Now, the clinical trials, which occur in 3 separate sequential phases, it takes years to finish, and on average only 1 in 20 drugs will perform well enough to even warrant entry into another complex set of process for FDA approval. Therefore, the best bet to find a treatment for an unknown human disease isn't to create one optimized potential drug, but 10 to 20. And out of these 10 to 20, you can actually expect that only one drug that will be approved for this unknown human disease. Here is where the biggest portion of the here it is where the biggest portion of the $2.6 billion that price tags come from. When you talk about these clinical trials, so most of the money out of this $2.6 billion that is spent on this particular clinical trials. Now, this is actually a diagram showing you the different phases of the drug discovery. The first stage, that is known as the preclinical, and as I've told you, when you perform the experiment in the lab, like in the petri dishes, or when you perform the experiments in the cells, the living cells, which are actually the diseased cells for that particular disease, or when you are performing experiments in the model organism, that actually comprise the preclinical stage of the drug development. When a lead compound or when a drug show good results in the preclinical settings, then that actually is approved for testing in the humans, and that enters into the clinical trials. Now, the clinical trials, that is actually divided into 4 phases. Now, in the first phase, as you can see, only a limited number of the participants that are, you can say, actually utilized, or they are actually allowed to participate in the study. So 20 to 80 participants, they are actually there in the phase 1, and they are actually tested for the effectiveness of the particular drug. When a particular drug that passes the phase 1, then they enter into the phase 2, where the human beings, they are again the volunteers, they are utilized, but here the number of participants that is increasing, that is between 100 and 30. In the phase 3, again you are increasing the number of the participants from like 1,000 to 3,000. Now, when a drug that clears all of these 3 phases of the clinical trials, then the drug is submitted for FDA approval. Now, once a drug that have been cleared in all of these 3 phases, you have to submit a very complex, you can say, documentation system to the FDA, where the FDA is going to review the safety and the effectiveness of that particular drug. They will be confirming the safety and the effectiveness of that particular drug. And once the FDA review committee pass the or confirm the safety and effectiveness of the particular drug, the drug is then approved to be used for the general public, where you can actually will be having like 1,000 plus participants, because the drug that will then be available in the market to be used by the human beings for the treatment of that particular unknown human disease. Now, what are the sources of the new drugs? Where can you actually find the new drugs? The sources of the new drugs, new drugs can come from the animal. For example, captopril is actually a drug that is derived from a poisonous snake, which is known as the Bothrops jararaca. And this captopril is actually used to treat high blood pressure and the heart failure. So animal can be the sources of the new drugs. So this is just one of the example that animals can be a source of the new drug. You can actually have new drugs from the plants, like quinine, which is actually an anti-malaria drug that has been derived from the cinchona plant. The microorganisms can be a source of the new drug, like penicillin, the famous penicillin that was obtained from the Penicillium notatum. You can also make new drugs by the chemical synthesis. For example, the Taxol, which was initially derived or extracted from the view plant, which is anti-cancerous in nature, that can now be chemically synthesized. You can actually go for molecular modification of the existing drugs to make new drugs. And in the molecular modification of the existing drug, what you do is that you add different functional groups that can be added to existing drugs to enhance their activity. You can have new drugs from biotechnology, for example the monoclonal antibodies used to treat rheumatoid arthritis that has been made by the process of the biotechnology by using the technology of the monoclonal antibodies production, and this is known as the adalimumab. Now, who are the contributors to the new drugs? A lot of individuals that are involved actually in the process of the drug development: the organic, the physical, the analytical chemist, the biochemist, the molecular biologist, the biotechnologist, bacteriologist, microbiologist, pharmacologist, immunologist, toxicologist, the hematologist, the biostatisticians, the marketing experts, and many more that actually contribute to the development of the new drug. Now, if you like the video, please subscribe to my channel, hit the like button and share it with your friends. And in the next part of this video, I'll be focusing on some of the important terms that are used to understand the concept of the drug discovery.